Okay, so the structure of the DNA and the way in which it codes for protein synthesis was worked out during the 1950s and 1960s. Since then, this knowledge has developed so much that we can change the DNA in a cell and thereby change the proteins which that cell can synthesize. Not only that, but we can sequence the nucleotides in the DNA and compare nucleotide sequences in different organisms. Now, what does DNA sequencing means? It means simply finding out the nucleotides of a DNA. By nucleotides, I mean that what type of bases they contain in the DNA, like adenine, guanine, cytosine, like this. It could be a sequence. So, gene sequencing is the process in which the order of nitrogenous bases in the DNA is identified. Okay. It is also possible to carry out genetic tests to see if people are carriers of genetic diseases, and in few use gene therapy to treat those who have these diseases. Okay, so gene technology has brought huge benefits to many people, but many have consequences that we cannot foresee. These technologies raise social, economical, and ethical issues that we must confront. Okay, so genetic engineering. The aim of genetic engineering is to remove a gene or genes from one organism and transfer it into another so that that gene is expressed in its new host. Say, for example, one organism which cannot protect one organism, or maybe a cell, or maybe a bacteria or virus, or say for example bacteria X cannot protect itself from bacteria A, but bacteria Y have a gene that produces a protein which kills bacteria A, so it is protected. Now if you transfer the gene that is responsible for making the protein which destroys bacteria A into the gene of bacteria X, which do not have protection from bacteria A, then bacteria X can also produce that protein just like bacteria Y, and so bacteria X will be protected from bacteria A. Okay. The DNA that has been altered by this process and which now contains lengths of nucleotides from two different organisms is called recombinant DNA. So if in the DNA of bacteria X I put the gene responsible for producing that protein or that substance into the gene of bacteria X, then that DNA of bacteria X will be called recombinant DNA because it is combined with the DNA with the gene or the DNA of another organism. The organism which now expresses the new gene or genes is known as transgenic organism or genetically modified organism. So if I did that to bacteria X, then bacteria X is a genetically modified organism. Now recombinant DNA is made by joining pieces from two or more different sources. Just what I have explained. Now, genetic engineering provides a way of overcoming barriers to gene transfer between species. Usually between species gene cannot be transferred because species cannot different species cannot interbreed, and even if you breed, the gene that we want may not be present in the offspring, or indeed the genes are often taken from an organism in a different kingdom, such as bacteria genes and inserted into a plant, or a human gene inserted into a bacteria. It is also possible through gene technology, genetic engineering. Unlike selective breeding, where a whole set of genes are involved, genetic engineering often results in the transfer of a single gene, so we can be much more precise. We will look first at the general principles involved in genetic engineering and then at some of the techniques in more detail. Okay, so how is gene transfer possible? Let's see. An overview of gene transfer. There are many different ways in which a genetically modified organism may be produced, but these steps are essential. The gene that is required is identified. It may be cut from chromosomes, made from mRNA by reverse transcriptase, or synthesized from nucleotides. The gene can be identified from either chromosomes or made from mRNA by reverse transcriptase, because we know that messenger RNA is complementary to the DNA, and mRNA is produced by the process of transcription. Now if we reverse transcript the mRNA, we get the code of the DNA. And if we can more best is to be synthesized from nucleotides. We can identify which gene we need and the sequence of nucleotides, sequence of nitrogenous bases. So we can there are also many free nucleotides. We can synthesize the gene, the sequence of the gene that we need to make the specific protein by synthesizing from nucleotides. Very advanced process. Multiple copies of the genes are made using the technique known as polymerase chain reaction. So polymerase chain reaction is the process in which a gene can be cloned and multiple copies of it can be made. This process is called polymerase chain reaction. The gene is inserted into a vector which delivers the gene into the cell of the organism. Examples of vectors are plasmids, viruses, and liposomes. Now the gene, we cannot simply put the gene in the new organism that we want to put. We need a vector. A vector is something which carries the gene which we want from one organism to the gene into the other organism. So always a vector is needed. Now vector could be plasmid. We know what is a plasmid. We can insert the new gene into the plasmid and insert the plasmid into the bacteria or the organism that we want to modify. It could be viruses and it could be liposomes. Now what are liposomes? Liposomes are spherical sacs of phospholipids. Inside the spherical sac, the gene can be inserted and the liposome can be inserted into the cell of the organism. The vector takes the gene into the cells. Yes, the cells that have the new gene are identified and clones. Now not all the cells are going to take in the gene. We will look at the process in great detail, but understand this, that not all the cells will take the gene. Few cells or few organisms will take the gene. How is it identified? First of all, say for example, if we put a marker on the gene which we have put on the vector, and then after that if we try to identify the gene, we can see that which organism has the marker. The organism which has the marker, obviously that cell or organism has taken in the gene which we wanted to put in, and then it is identified, and then the cell of our choice or the organism of our choice is cloned so that many copies of it is available. We will see how genetic engineering has helped us in many ways. To perform these tests, the genetic engineer needs a toolkit consisting of enzymes such as restriction endonuclease, ligase, and reverse transcriptase. Enzymes, of course. We know that let me tell you that reverse transcriptase is an enzyme responsible for reverse transcription. If we have taken the gene by the process of reverse transcription, then we need the enzyme reverse transcriptase. And now restriction endonucleases are the enzymes that cut the specific part of a gene, and ligase enzymes adds or makes bonds to the genes. Say for example, in the plasmid, we will use restriction endonucleases to cut the gene from the organism we want the gene. We use the same restriction endonucleases so that the gene is cut into the plasmid, the vector. We can cut the gene of the plasmid. Then there will be a space where we can put the gene of choice. And then when we put the gene of choice to the plasmid, we need something to make it into one, or to add it like a glue. That the enzyme responsible for forming the bonds again in the plasmid is ligase. Okay, so endonuclease, you have understood. Vectors including plasmids and viruses. Genes coding for easily identifiable substances that can be used as markers. Say for example, we have put a marker in the gene of our choice that lights green color in ultraviolet ray. When we have put the gene into the vector, and the vector puts the gene into the organism or the cells, not all the cells have taken in the gene. So how do we identify from the culture? of cells or organism, that which organism or cell have taken in the new gene. We again shine it with ultraviolet ray, and the gene which we took from the organism of choice lights green again, and then we understand that these cells or these organisms has taken the gene. Then we take only those cells or those organisms and clone it.